Okay, now move on to something a little different, more focused on genetics and fitting models and model selection in genetics. And first, I'd like to welcome somebody not a million miles away from my office, Doug Speed, who's working with me at University College in London, who's going to tell us about heritability estimation from genome-wide SNPs.
Thanks very much, and thanks for a chance to talk here. So as you see from the title, I'll be talking about estimating heritability from genome-wide SNP data. And as I mentioned in the abstract, maybe on the face of this is far removed from model selection, in the sense that heritability analysis is looking at the total contribution of a large number of variables. Yeah. So maybe on the face of this looks like a completely different problem, heritability analysis, in that we're looking at the contribution of very many variables, but hopefully, as I'll touch on, it can be used for model selection and variable selection in some ways. So the catalyst for this talk was a paper which is about 2 years old now by Peter Vischer and Jiang Yang, who are based in Queensland in Australia. And their key result, they applied the mixed model analysis to human height. And the result here was that they deduced that using a model which allowed the contribution of all the SNPs on the genotyping array, they could explain 45% of the variance of human height. And just to put this into perspective, until then, association studies had managed to explain only about 5% of the variance. So this was about a 10-fold improvement. And so this paper was really embraced by the association study world. And most of you are probably familiar with this idea of missing heritability in the discussion and argument. And so a lot of people have suggested that maybe the reason that for these very heritable traits, so for complex traits, we know there's a very large genetic component. The reason why association studies have done so poorly in explaining the variation and finding causal variance, well, one argument is it might be due to rare variants, which a genome-wide association study, the genotyping arrays typically focus on common variants. So while they do a very good job at tagging all the common variation, or most of it in the genome, they don't focus on rare variants, by which I mean less than, say, 1%. But by contrast, this paper of Peter Vischer and his group, where they deduced that 45% of the variance of height could be explained by these common SNPs. And so the association study crowd have really made use of this to say association studies are not dead. It's simply a power issue, that there are very many causal variants which are common, minor allele frequency. It's just that their effect sizes are too small to be picked up from a standard association study. And since this was published about 2 years ago, pretty much not a single month goes past without them publishing another Nature paper or American Journal paper. And so here are just a few. So for example, the top left, GCTA, is the software they published for doing this mixed model analysis. And then they released, although it's designed for a quantitative trait, which height is, they showed how it could readily be applied to binary traits, so for the case of disease. And then this third paper is on genomic partitioning, which I'll talk about a bit more in detail. But here the idea is that you can consider the heritability of all SNPs, so how much variance all SNPs explain, but also you can break this down into tranches, and that's where the model selection, variable subset selection comes in, that you can evaluate the heritability of variance explained by a particular subset. And in the same fashion, it's been applied countless times. So here are just 3 examples where it's applied to schizophrenia, arthritis, and and intelligence. And in all of these cases, they have the same conclusion, that association studies are doing very badly to explain the variance of these known to be very heritable traits. But using this mixed model analysis, they find a many-fold improvement. And they've also used this to argue that these traits are highly polygenic. And this is partly the way they do this genomic partitioning. They break it down and consider the heritability from a particular chromosome, for example, and they show that the contribution to these traits is sourced from every single chromosome almost equally in proportion to length. And so that's how they deduce that these traits are very polygenic with a lot of causal variance. It's fair to say there has been a bit of suspicion of the method. Maybe not so much has been published, but here is one criticism by Browning and Browning, and they used an example which I'll talk about later, of how the method might be vulnerable to population differences between cases and controls. But otherwise, I think the methods have been well received and are being used quite widely. So just for clarity, this mixed model obtains estimates of heritability, but it might be easier to think of these estimates in terms of variance explained. So for example, you can then, these estimates are important because you can compare them to the trait's known heritability and see, well, what proportion of the heritability can we explain by the genotype SNPs? So using the example of height, it's well recognised that the total heritability is approximately 80%. So the fact that their paper suggested 45% of the variance could be explained by the genotype SNPs suggests that we can explain just over half the heritability. And I'll explain why there's this distinction between variance explained and total heritability. And then to touch on again this idea of genomic partitioning. So this is where they considered the heritability from particular subsets of SNPs. So the idea is that you can break down all the SNPs on the genome by a particular function, so maybe by minor allele frequency, or whether they're close to a gene or outside of a gene, or some particular functional property. And then you can assess the contribution from different subsets of SNPs. And the idea is that then you perhaps elucidate some ideas about the genomic architecture. And maybe it's best to give the example. So we applied this to our epilepsy dataset. And epilepsy, for those of you who've had any experience, if you take a Manhattan, it's flat as a pancake. There's nothing of interest there whatsoever. No large genome-wide hits except for maybe a few Mendelian causes. But the heritability of epilepsy is estimated to be perhaps 40%, and we found that maybe 30% could be explained by this genome-wide model using the mixed model analysis. And furthermore, when we broke down the SNPs into these 4 tranches, so we divided it into 4 sections, those with minor allele frequency less than 12%, the second quarter, the third quarter, the most common SNPs, and then assess the contribution of the heritability from each of these, we find that all of it's contained within the first 3 quarters. So this might then give us the impression that for epilepsy, the causal variants are predominantly slightly more rare. So maybe there's not so much interest in the most common SNPs. But all of these deductions we make from, in terms of heritability analysis and genomic partitioning, all depend on the validity of the method, the mixed model method, and that's what we set out to investigate. So first of all, to explain the mixed model method from a mathematical point of view, I've got one slide. And so it all starts from this linear model. So here we have the standard linear model. So we have our phenotypic value, y. Then we have here, these are the causal SNPs, z, and they have been standardized, which I'll talk about a bit later. And so we assume that the contribution of the causal SNPs each has its own effect size beta, and these add up with an inset term. Add to this the random error of the noise, and that's how you get your response. And so if we're to take perhaps a Bayesian point of view and assign a normal distribution to each of these effect sizes, notice they have common variance, sigma g. I've divided by n to make the maths a bit easier, but each of these have the same variance. These effect sizes are assigned to the standardized causal SNP values, and if similarly we assign a normal distribution to the noise, then we get this equation. And essentially here the variance of the phenotype can be broken down into this matrix G, which here is Z transpose, which is a correlation matrix of the causal SNPs, and then this second matrix, this identity matrix, which corresponds to the noise. And so the idea of heritability analysis is to obtain estimates of, well, relative values of sigma G, which is a component of genetic effect, and sigma E, which is the noise term. But the thing is, because we of course don't have the luxury of knowing which are the causal SNPs, then in place of G, this matrix over relating this over causal SNPs, we replace it, typically the common sense choice is to use A, the matrix over all the type SNPs. So again, these are the type SNPs X, and these are standardized. So that's the fudge we use. We don't know the causal SNPs, so instead we consider correlations over all the SNPs. So that's a mathematical description, so maybe a bit more intuitive. Suppose we have our phenotype. Here it's very simple, it's either 1s or minus 1s for different individuals. And this is for phenotypic correlation. So you can see when the individuals have the same phenotypes here or here, then their correlation is 1. And when they have discordant phenotypes, it's -1. And so the idea is we then are looking to divide the variance of this phenotype into these two matrices. Here A is the relatedness matrix over all SNPs, which is very similar to a pedigree matrix. So, for example, each individual is relatedness on average 1 with itself. You get a phase of 0.5 when you have full SNPs and so on. So we're looking to divide the variance between this relatedness matrix and the noise matrix. So if we notice that the individuals with the same phenotype, these ones in red, are on average more related, then that implies there's a large genetic contribution to this particular trait. And so we divide the variance in these two ratios, and then our total heritability estimate is a fraction of genetic variance over the total variance. And I talked about genomic partitioning, so this is where we extend this case to, rather than just having a noise matrix and one correlation matrix, we then consider a correlation matrix over, say, low minor allele frequency SNPs and a second matrix over medium minor allele frequency and so on. So we can extend this to multiple correlation matrices, and then we can still, we're dividing this, not this variance between a lot of correlation matrices. So that's the idea. So these slides are intended to show the difference between what we're doing to traditional heritability analysis, because it has been used for many decades in animal models when you have family data. But the key insight of Fisher and Yang was to apply this method to unrelated individuals. So when you go back to the traditional method of using related individuals, where you have a lot of concordance, a lot of genome sharing between particular pairs. So this is quite a pictorial representation, but here we have a section of the genome of two individuals, and this red bar indicates that they're concordant for these genomic regions. And bear in mind, they might still share base pairs, of course, on these other regions, just by chance, but these are, for example, regions they've inherited from a recent common ancestor. This third line represents all the variants. So of course we don't know where all these all the variants are in the genome. Instead, we only type a subset of these. And if you remember, for mixed model analysis, we are trying to estimate the correlation across the causal SNPs. So we don't know which of these are causal. But for example, here are 7 SNPs, 7 variants in this region which is shared between the two individuals. So we can appreciate that even though we only see 2 of these type SNPs, the relationship, the correlation across these type SNPs would be a good indicator of relationship across all these 5 other SNPs. And in that sense, because we have long regions of concordance, even though we only type a subset of SNPs, when we have related individuals, we're able to capture the correlation across more variants than just those we type. And in that sense, when we have related individuals, our heritability estimates are heritability estimates of the entire trait. And so for height with related individuals, we'd expect to get 80% consistently. The difference in using, as it were, unrelated individuals is that you have these 2 individuals which are very, very distantly related, and they share only very small regions, and perhaps this is only because of linkage equilibrium rather than genome sharing. And in this sense, when you calculate the relatedness over a type SNP, so the ones we observe, it will only represent the relatedness over variants which have been observed, so there's not this long genome sharing. So in this sense, when we apply heritability analysis to unrelated individuals, our estimates are focused on the type SNPs. So our estimate is for variance explained by the type SNPs only, rather than an estimate of the total heritability of the trait. And this picture shows this effect. So along the x-axis, this indicates the proportion of the genome we've used to calculate the relatedness matrix from. So here we've simulated a response with 80% heritability, and we've drawn the causal SNPs from throughout the genome. So when we calculate relatedness over only a quarter, on average we will only pick up a quarter of the causal SNPs. So we'd expect to only get a heritability estimate of a quarter of 80%. And here we can see that for red individuals, so these colours indicate the amount of relatedness we have in the sample. On the left-hand side, our estimates consistently come close to 20%, as we'd expect. But when we introduce related individuals into a sample, we can see that even though we're only capturing a quarter of the causal SNPs, we're actually getting heritability estimates higher than that quarter of the heritability. And then as we increase the proportion of the genome we genotype, across which we calculate relatedness, well, refraction increases as we'd expect. And when we get to the point that we're actually calculating relatedness over all the SNPs, it makes no difference whether we have related or unrelated samples. We still manage to recover the complete heritability because we've considered all the causal SNPs. The only difference is, with high relatedness, we get more precise, accurate estimates. So, as I explained the mixed model, I pointed out there were three implicit assumptions which were the first three things we tested. So the first one was the fact that we would like to calculate relatedness over the causal SNPs, but instead we use relatedness over all the SNPs. And because we assign this normal distribution of effect size prior to the SNPs, the fact that we do this fudge of using all the SNPs implicitly supposes that actually every SNP is causal for the phenotype. And so we have this model where every SNP is assumed to contribute to a response, even though, of course, we know that's very unlikely to be the case. We might have a few thousand causal SNPs, but probably not every single SNP contributing. So the question is this a problem? And first of all, I'll explain this diagram because I'll show a few of these throughout the talk. So here the idea is we took 5,000 individuals from a Wellcome Trust, so UK samples. We concentrated on the first two chromosomes, so about 80,000 SNPs, just for clarity. And we simulated responses. So these green responses we simulated with 80% heritability. The red ones have 50% heritability. So the method in which we used to test the mixed model was to see how well these estimates, how well the green box plots fell close to this green line, and how well the red ones fell close to the red line. That's suggesting the mixed model is producing accurate estimates. So here we've tested what happens when we reduce the number of causal SNPs we simulate as causal. So in this right column, we have all the SNPs causal, so all 80,000, which is the implicit assumption of our analysis. And we see that our heritability estimates are dead on what we would expect. But as we reduce the number of SNPs, although for 2,100 causal SNPs, it makes very little difference. We see as we reduce to, say, only 20 of SNPs or only 5 of SNPs are causal, the variance of our heritability estimates increase. But the key thing to notice is they remain perhaps unbiased, it would appear. So they're still consistent with the heritability simulated. And this is quite staggering. We've found that even when you only have one SNP as causal, and you calculate the relatedness matrix here over all 80,000 SNPs, you can still pick up the effect of this one causal SNP using mixed model analysis. So in this sense, we have this assumption that every SNP is causal, but it doesn't seem to matter if that's not the case. The second assumption is that I mentioned we standardize for genotypes by subtracting the mean and divided by the standard deviation. But we have this prior, this Gaussian prior for the standardized SNPs, sorry, this should be a sigma G. And this is for the standardized SNPs with a constant variance of effect size prior. And this implies a prior on the raw SNP effect size. And so this implies that the effect size of the variance of the raw effect size is proportional to 1 over standard deviation. So to try to make this more clear, well, under Hardy-Weinberg equilibrium, we'd expect a SNP to have variance 2p 1 minus p, where p is minor allele frequency. So what this assumption and standardization does is it assumes that the effect size is proportional to 1 over 1 minus p, the square root of that. So many people would consider this a sensible property. A lot of people think that rarer SNPs are more likely to have larger effect sizes. But to put a few numbers, if we have a SNP with minor allele frequency 0.5, its variance would be 0.25. And that would be approximately 25 times higher than a variant which has 1% minor allele frequency. So this specific relationship is assuming that a SNP which is a 1% frequency has 5 times on average the effect of a SNP which is 50% frequency. So why should we use this value 5 times? Why not 2 times? Why not 10 times? So one of the things we've asked is, well, does it matter that we use this very particular relationship? And on the next slide, I'll use this form. So to test different relationships, I've just written this slightly different, but with to the power of alpha divided by 2. So the idea here is that when alpha equals -1, this is a standard assumption where you divide by for standard deviation, but we can consider different values of alpha, which I'll show you on the next slide. So here we have two values. Alpha 1 is the relationship between minor allele frequency and variance size we've assumed when we've simulated the responses. Alpha 2 is the relationship we've assumed when we're doing the analysis. So for example, this block of 4 pairs of box plots, we've analysed the data assuming alpha 2 equals minus 2. So if you remember, alpha minus 1 is a standard assumption. As we go to this left side, this assumes that rarer SNPs have an even greater effect. By contrast, alpha equals 0, this assumes that the effect size is independent of minor allele frequency. And on this right-hand side, this is where we assume that actually common SNPs, more common they are, they have larger effects. So we've tried a spectrum of different situations. And what we first notice is, as you'd expect, when alpha 1 equals alpha 2, so we analyse using the same assumption that we've simulated, which are these grey boxes here, we obtain unbiased estimates. So for example, here's alpha minus 1, alpha equals 0, and alpha equals plus 1. But of course, we don't have the luxury of knowing what the underlying relationship, what the underlying system is. But one thing we can ask is, well, which is the best block to use for analysis if we're agnostic of what the true relationship is? And we find that when we have alpha equals minus 1, which is the typical approach used, well, this turns out to be the most robust in the sense that even when we violate this relationship, the estimates still seem closest to the true value. So they are best if the true relationship is minus 1, but when we go either side, we don't seem to lose too much. So again, this was the second assumption, this specific assumption which a mixed model makes, but it seems that it's not a problem that it makes that assumption even if it's not true. The third thing we considered was the fact that we assume Gaussian effect sizes, and these are often criticised in association studies because they're very thin-tailed. So here we have a plot of the effect sizes, and these red bars indicate the 99.99 quartiles of this particular Gaussian distribution. So here we see there's a 1 in 10,000 chance, this is assuming of an effect size being greater than 0.1. But we know this is not really the case because there's so many association studies where we've had very large effect sizes, very large odds ratios. This probably isn't a realistic distribution of effect sizes. And so to contrast, we've used the normal exponential gamma, and this is a distribution which potentially has thicker tails. It has a shape parameter, which I won't go into detail, but here, when the shape parameter is relatively high, which 10 is, it resembles a double exponential distribution, and so maybe the tails are approximately equally thick. But as we decrease this shape parameter, it becomes more spread out. So here we see there's now these quantiles, there's a 1 in 10,000 chance of being greater than 0.3. For this value of 1, well, the quantiles are so spread out they fall off this plot. So it's a much more thicker tail distribution. And so we tested simulating responses using these underlying distributions of effect sizes, but yet analysing it using the standard assumption of Gaussian. And so here, when we simulate with Gaussian, yes, the method performs very well as is expected, but we're surprised to see that even though we're using radically different distributions to simulate effect sizes from, even though we're analysing with the Gaussian assumption, the mixed model method still seems to be reasonably consistent. So that's the third test which the method seems to pass. However, by contrast, we've noticed that the method is sensitive to linkage to equilibrium. And to explain how we notice this problem, the method used is known as REML, which I won't go into details, but this is how we obtain this estimate of heritability. But bear in mind that heritability is bounded on 0, 1. So this method gives you a standard error, but this assumes a normal distribution. So if you get an estimate of, say, 0.5, it might say it's a confidence interval between 40 and 60%. But of course, this normal distribution is inappropriate when you're trying to estimate very small values. And in particular, how would you estimate whether a value is significantly different from 0? Because there, it's certainly not a symmetric confidence interval. And so one idea we had was to artificially inflate the heritability of our real data, x, by a value which we specified. So for example, we increased it by 50%, and we did that by adding on some artificial SNPs which are independently generated just in Hardy-Weinberg equilibrium. And then we also used a new phenotype. And as I say, we knew that the estimate, the heritability estimate here, would be 50% higher than analysing it using the original data. So the idea is if we want to test whether the original heritability was 0, we can instead test whether this new data gives an estimate significantly different from 50%. And that was our idea. But when we did this test, we were surprised to find that the heritability estimates on this revised data were consistently less than 50%. In fact, they were more like 10 or 15%. And we weren't sure why that was until we realised that the signal from these SNPs we were adding on, because we had generated them in Hardy-Weinberg equilibrium, they're all independent, their signal was being underrepresented in this correlation matrix. So to try and explain why, this gives an idea of what the correlation matrix does. It computes each value, compares, so AI prime compares the concordance between two genomes. So here's individual 1 SNPs, individual 2 SNPs, and it moves along, saying, well, when the SNPs are different, you get a negative value. When they're the same, it adds on a small value, and so on, and it moves down. When it encounters, you can appreciate when it encounters an area of high LD, you have a lot of very concordant values. So this region here counts 3 or 4 times towards this estimate of A. So this area of LD has an inflated effect, and we find that that carries over into the heritability estimates. Just to pictorially show the difference as we move along the chromosome, the whole genome. Here, this y-axis shows a measure of tagging. So we consider a statistic which is the correlation of each SNP with itself, plus the correlation squared with all SNPs in a particular window. So you see that it's far from uniform as we move across the genome. So for example, we have some SNPs. The medium SNP, its signal is tagged 58 times on average. So it has its signal is replicated on average 58 times. But here, for example, is chromosome 6, the MHC region, is over 600 times replicated. But conversely, we have other SNPs which are actually represented only once, essentially, because they're so independent in their particular window. And so that causes problems for the heritability analysis. So here we've chosen the causal SNPs varying the degree of tagging. So when we pick all the causal SNPs to be very poorly tagged, we find these estimates are much reduced. So for example, we've estimated 80% heritability, but our result is close to 45% heritability when they're very poorly tagged. And conversely, when the causal SNPs are very well tagged, we overestimate heritability. And this carries over to minor allele frequency, because as you can appreciate, rarer SNPs on average worse tagged. So this has implications. If we use heritability analysis for rare variant diseases, well, these rare variants will on average be poorly tagged. And so this heritability will underestimate. So here, the causal SNPs are less than 10%. We find that we are about 20–25% underestimate the heritability. So just to point out that there's 2 different issues. One is the fact that some causal variants will not be tagged at all. We won't have considered them. We won't even be in type SNPs which tag them or be in regions. And so that's something we can't do anything about. If a causal SNP has not been tagged in any essence, we can't estimate its heritability. However, what we're referring to is the fact that some causal SNPs are tagged, but some are tagged better than others and some are tagged worse than others, and this uneven tagging is what's causing the problems. Now, anyone who's done an association study would have encountered this problem when they're constructing principal components analysis to identify population structure, and there the solution is simply to prune the SNPs using a correlation squared. But you have to appreciate for heritability analysis, if you discard 90% of the SNPs, for example, that's going to decimate most of the signal, because we're looking at very small contributions, fractions of percent from each SNP. And so if you remove 90% of the SNPs, you'll probably lose on average 90% of your heritability. And so that's not a solution. But what our solution was instead was to construct these weights for each SNP, so that when we calculate this correlation matrix, we then upweight the SNPs which are poorly tagged and downweight those which are well tagged. So to give the intuition, suppose you had 2 SNPs which were identical, but they're in linkage equilibrium with all other SNPs, then you'd expect these to get weighted in 0.5. And that's what happens, except weighting also considers when you have impartial tagging and lots of SNPs in different correlations with each other. And this just shows that this is the tagging before we apply our weightings. And we can see that after we apply our weightings, the average variance of each independent signal is approximately 1. It's not an exact method, it's linear optimization, which is why we do get these discrepancies, but it produces a much flatter graph. And we find that when we apply our weighted correlation matrix, these biases we used to find based on the tagging of causal SNPs almost seem to disappear. And just to return to the polygenic, where we vary the number of causal SNPs. Previously, we saw that as we reduced the number of causal SNPs, the variance of the estimates increased significantly. And now that's perhaps easy to explain, because when we have only one causal SNP, well, half the time this causal SNP is likely to be undertagged, and then we're going to underestimate and get a much lower estimate of heritability, even though the method is working. Half the time it would be overtagged, and we're overestimating heritability, hence the very large spread we see. Whereas when you do this weighted correlation adjustment, it doesn't matter, because now the causal frequency doesn't affect the estimate, so we get much better precision. This was just going back to when we varied effect sizes, so it's still robust to violations of the effect size of a Gaussian distribution. And similarly, this distribution relationship between minor allele frequency, so we still see that using this alpha equals -1 appears to be the best assumption to make. So one of the reasons I mentioned this Browning and Browning paper criticised the heritability analysis, because it suggested that it was affected by population structure. And ideally we should use a homogeneous population so that these are not an issue. Of course, that's never going to be the case, and we try and correct for it by using principal components. In a sense, this is very similar. Population structure is synonymous with relatedness. It's all a form of cryptic relatedness. So we have the same problem if we actually have slight relatedness, which is inevitable in our sample. We might also get this inflation due to population structure or relatedness. I'll focus on population structure. So this paper, they performed a pseudo case control study, and what they found by pitching one Wellcome Trust control data set against the other, you'd hope that you would explain a heritability of 0, but in fact they found significantly non-zero estimates, and therefore they concluded that the method was unreliable. So just to show what this study did, they took a Wellcome Trust control as approximately 3,000, and there's broad geographic regions for each of these. They picked out the Scottish and the Welsh ones, and of the English samples, they decided 90% of these were controls, and of the Scottish Welsh ones, they decided 90% of those were cases. So this case control indicator was highly correlated with whether you were English or not. They actually used the word disease, so it's quite a trivial example, but just for my fun.
So
this is
being English was diseased, is it?
No, quite the opposite. They got it right. Yeah, so if you're English, you're 90% likely to be normal, but if you're Scottish, there's very little hope. Yeah, I wouldn't laugh. This is not something we can treat, so it's very serious. And we replicated this, and we found the same problem, that when we had this ascertainment of cases and controls based on geographic region, these are heritability analysis. We'd hope them to be 0, but instead we found they were 20, 30%. So this does suggest there's a problem with this method. So one of the things we've done is looked whether there's a solution, and this is actually inspired by one of the papers of Yang and Vischer. And the idea is that if you contribute for heritability from the whole chromosome, so you get an estimate which is the whole heritability plus this contribution from population, this inflation P. Whereas if you contribute for heritability from just the first half, so from chromosomes 1 to 8, you get the heritability from the left half, plus you get this same inflation. And the reason for this is that population is a very broad thing. So if you have 400,000 SNPs, the whole genome, or just 200,000, we'd expect the contribution from population to be the same. And in this sense, by performing this heritability analysis 3 times, you can assess what the inflation due to population is. So the black lines are now covered. This was the estimates from the whole genome. And we can see that when we estimate from just the left half, the red line, the heritability estimates are almost the same. If it was true heritability, we'd expect only to pick up half of the heritability from using the left half. And same from the right half, we'd expect to only pick up half of the true heritability. But we can see that because all figures are almost identical, then population stratification for inflation must be contributed to these results. And you can imagine, therefore, we can correct for the contribution, the inflation due to population stratification. We can calculate how much it is. It's the difference between the estimate of the left plus the right minus the whole, and we can calculate this corrected estimate. So in a sense, although they were correct to point out that there is a problem due to population differences, it seems that we have a test to see whether this is actually affecting the results. So we shouldn't use this to cast doubt on any of the results, provided we check. Actually, this slide is asking whether you can still, if you do have this problem of population inflation, whether you can then correct the estimates. I'm just going to skip over this because I'm getting a bit late. But the idea is whether, even though we know there is population inflation, can we correct the estimates by guessing how much the inflation was? And in this case, I simulated 50% true heritability and tried to cancel out what we knew to be population. And we were getting estimates of maybe 30%. So maybe this suggests that although we can tell whether there is inflation due to population structure for very large scale population inflation, perhaps we can't fully correct for it, but it's still something we can check to see whether this is an issue. So a second criticism which we've noticed is the heritability analysis is very susceptible to genotyping error. And this is much more of a problem than in an association study, because there you're just looking for the very top associations, so you're just considering each SNP one at a time. Whereas heritability analysis allows contribution from every SNP, so these errors will accumulate over every SNP typed. And one of the things you could do in an association study is go back and check your top results and see, well, is this a genotyping error? Look at the cluster plots. But for heritability analysis, you're getting these global estimates. You can't then go back to your results and check. So you have to be very careful. So it's recommended that you use very high and very stringent quality control checks. But we're trying to see whether we can come up with a way to test the contribution of genotyping error in the same way we can test the contribution of population inflation. And this is a very similar idea, except here we calculate heritability over whole and compare this to the heritability over all the odd SNPs and then over all the even SNPs. And we expect the heritability contribution from the odd SNP, so every other SNP, to be very close to that of the total heritability, this factor R, which we can estimate a value for R. I won't go into the details, but essentially we can obtain a bound on what the maximum possible heritability error is if we thought that R was 1, so where the odd SNPs perfectly capture the variance of the even SNPs. And we can get realistic bounds to see what the contribution of genotyping error. So although this is a very valid criticism of the method and a caveat, we think we can now test to see whether our estimates are inflated by genotyping error. So just briefly, I'm going to show the application of the methods. First of all, we were very kindly helped by a member of Peter Fisher's lab in Queensland, Gibb, who applied our method remotely. Because we couldn't get access to the data, he wrote some scripts to apply our weighted version to their height data. And first of all, we replicated their results at 45% variance explained. And then we used our weighted correlation matrix, and we were hoping to get this great improvement. And we did. We got 46%. Fantastic. So not really any different. And one of the things they mentioned in their paper was they suggested evidence, a bit small to read here, but they did notice this effect of poor tagging of rare variants. And what they suggested, they did some simulation studies and suggested that if all their causal variants were minor allele frequency less than 10%, then that would explain why their estimate of 45% was so, even though all the causal SNPs were tagged. So if they had typed all the causal SNPs, and so there was 80% true heritability, they hypothesized that maybe because they were rare variants, their method was underestimating it. But I guess our result, because our method is not susceptible to tagging of causal SNPs, it seems to give unbiased estimates regardless, then we can perhaps rule out this hypothesis that for height, a lot of the causal SNPs, while common, are of lower minor allele frequency, so there seems to be no evidence from our results. And just finally, we applied our method to all the Wellcome Trust data sets. This is from the very first study, so it's relatively small sizes. But just to pick out a few of interest, so we applied the standard method and see when our weighted method gives the largest gain or loss. And so, for example, for hypertension and type 2 diabetes, we find that we get increased heritability estimates from using our method. And if we examine Manhattan plots, well, maybe hypertension is one of these nice examples where there's no genome-wide significant results, except perhaps one here, but yet we can still explain a large proportion of the heritability using this Omni model. And maybe the fact that our method has increased the estimate suggests that it is the cause of SNPs on average more rare. By contrast, for these two traits, arthritis and type 1 diabetes, our weighted correction method reduced the estimates of the standard method. And here it's perhaps very easy to see why, because both of these have their strongest causal variants with odds ratios of maybe 3 and 5 in the MHC region of chromosome 6, which, as we know, is a very well tagged, very high LD region, and therefore it's understandable that the standard method would have overestimated the variance. But we can consider just for chromosome 6 heritability, and we see that for a standard method we get particular values 19 and 37%, but when we apply the weighted values these decrease a bit, and this accounts for maybe half of the difference for type 1 diabetes. So just for conclusions, we think we've tested the mixed model quite thoroughly, and while it seems to be very robust to many of the implicit assumptions, if you violate them, we did find it was sensitive to LD, but hopefully this weighted version will correct for these biases which are introduced. We feel we've got these tests now for population inflation and genotyping error inflation, which will hopefully make us more certain of the answers. And actually, my personal view is, in a sense, coming back to a bit of model selection, I think that this heritability analysis will be the best solution to pathway analysis. This is where you're trying to see whether a set of genes are associated, so you can't find any particular variant. You want to ask whether a particular set of genes are associated with a phenotype. And there the problems you have with standard methods are genes are of different lengths. Well, this heritability analysis perfectly accommodates for different lengths automatically. You have different levels of LD within genes where this accommodates for it. And because you can do this genomic partitioning, you can compare one pathway to a second pathway to a third, so you get this competitive model. So the fact that we can now get more estimated heritability analysis, if we have large sample sizes, maybe pathway analysis using this method might be a possibility. And then of course the next step is trying to then convert these results, these good heritability estimates, into prediction models. So the results do provide genetic values for each individual. And so heritability analysis for mixed model is in a sense Bayesian to try and get these SNP loadings. If we want a prediction model, so saying, okay, we want to predict whether one individual what his risk for epilepsy is. At the moment we have a model with 500,000 SNPs and effect sizes. We can go along on the test data set and then try and calculate the new test individual's risk score. But at the moment we have to revert to classical methods to calculate these SNP loadings. So we're trying to see whether we can perhaps get an improvement by a pseudo-Bayesian version. Because one of the things we noticed, a different paper noticed, was even though you can explain 80% of the variance of height on a test data set, on the training data set, when you apply this to a test data set, you can only explain maybe 20%. So there's still this gulf between the two, the fitted and the test model. Finally, acknowledgement. So this is all work with David. And we do have a paper coming out, thanks to Vincent, Mike, Johnson, Gibran, and Will for my funding. And finally, just an advert, is anyone going to the Edinburgh conference in June? Because we have 3 of us are going, and we booked a 5-bedroom flat. If any 1 or 2 people want to join us, I think that's very important to point out at this point. Thanks very much.
One question is, are you going to the conference? But the second question is, would you share a flat with this man?
The other 2 people in this room.
Right, so time for a few questions. Yes, Robin.
When you introduce your weighting method, you put up two different arguments. You can go back
Yeah.
to you do 2 arguments for why you should want to do the weighting in terms of the problem with the looking. Can you just go back? So yeah, you put the other fantastic. Yeah, the problem is not something but heritability will recognize a contribution. I wonder if that was contradicted but when it went on later on to say you're using half the SNPs in the odd states and you use my argument is that you can get a slip effect out. You're not just getting the effect of that slip, but the slips around it.
That's correct. So David likes to view this that actually we're not typing any of the causal variants. They're all untyped, but we're only picking up a tagging of them. And it's correct to say that when we use just the odd snips, we capture maybe 94% of the variation of all the snips. So we do pick it up. But even when these snips are not typed, hopefully I can answer this question, even when the snips are not typed, we still, if an untyped causal variant is very well tagged, it will still inflate the estimate. So you can have an untyped variant, but its tagging is still too much. And so you still get an inflation, even though it's not being typed. So it's relevant to both the typed and the untyped variants. One of the problems is our method, because we only know the type snips, the weightings are constructed based on the type snips, so they equalise the variance of the typed signals. But what we've done by masking the odds and the evens is we've shown that our weightings seem to do a reasonably good job of equalising the linkage disequilibrium of the untyped odd snips, even though we've constructed weightings just on the even snips. Does that answer?
Yeah, can I just add,
Yeah.
When you look at a logarithmic analysis, is there anything one can do just on one analysis in terms of diagnostics to suggest what next analysis you might want to do, rather than go through all those wide range of analysis that you have suggested?
As in the different tests we suggest?
Well,
yeah. Well, hopefully we're trying to promote that this is the method to use in place of the original method, and it does have some drawbacks in terms of computational time. To calculate these weightings takes about as long as to calculate the relatedness matrix in the first place, so perhaps it doubles the time. But we'd like to argue that this is perhaps a preferable method to use, and then these two tests for population structure and genotype and error, each of those takes about the same time as doing the original analysis, so we are talking quite a bit of computational resources required.
Seems to me this is a terrific methodology, but it's a bit disappointing that the principal components don't get rid of the population admixture effect or whatever it is. I'm just trying to think what that's telling you about the nature of the genetic difference between England and Scotland. What I think that's telling you is that you haven't got a particular set of variants that are characteristic of being Scottish, but just that any two Scots probably have a common ancestor rather closer than the Scotsman and an Englishman. But who that common ancestor is differs from one pair of Scots to another. Is that correct?
I think that's very true. And also perhaps they picked a clever example, because you might expect a reasonably continuous gradient from Scotland to England, but they included the Welsh as well as the Scottish. So maybe principal components would only work if you had a continuous gradient between the Welsh and the Scottish and the English, so maybe that disrupted a bit.
Okay, we're running over time, but as organizer, you're allowed to ask a quick question.
So, I love your idea of using that to do some particle analysis, because that seems like a much more powerful way than anything I've seen before. What happens if you do a gene-based analysis? I mean, there was a question of combining multiple variants within a gene and ask whether you know more than single. Isn't it the right way to do a gene-based analysis for association?
We think so. So the caveat is with maybe 5,000 individuals, your standard error estimates are approximately 5%. So you have to have a sizable contribution. So at the moment you can look at wide tranches. So they've split the data into different chromosomes, so 22-fold. But for a gene pathway analysis at the moment, with a reasonable number of individuals, you will need pathways and genes which are maybe 2,000 to 5,000 SNPs to get a reasonable heritability estimate to come within a bound. But hopefully, as we increase the power with more samples and more accuracy.
I mean, the standard deviation estimate that's sort of spat out by the analysis seems to be the same whatever the estimate is, so you get a relatively high uncertainty attached to it, but it doesn't seem to reflect actually the precision on replicate experiments. So, we better move on. Thank you very much, Doug. And